Okay, I think we can get started. Hello, everyone. Thank you for joining the Division of Rheumatology Grand Rounds today. A couple of reminders before we start the session. This session is currently being recorded and it will be posted later on our website. There will be a Q&A session at the end, so please leave your put your questions in the Q&A feature so that we can moderate them easily. And now, Dr. Mustelin, I will let you introduce our speaker for today.
Thank you, Danielle. Good morning, everyone. It's a really great pleasure and honor to introduce Professor Bill Robinson this morning. So Dr. Robinson got his undergraduate training at Stanford, did a PhD there in '95 and an MD degree in '96. And then moved not very far to UCSF to do a residency in internal medicine on a research track as an MD PhD. And then came back to Stanford in '98 to do a fellowship in rheumatology. He became an assistant professor there in 2003 and promoted to associate professor in 2010 and full professor in 2018. And in 2019, he became the chief of the Division of Immunology and Rheumatology, also at Stanford. From 2003, he's worked as a staff physician at the VA in Palo Alto as his clinical site. So Dr. Robinson has been incredibly active in the rheumatology field, in the ACR, in the Arthritis Foundation. He's been the editor of many rheumatological journals. He's involved in planning meetings and in committees. He's been awarded many prizes and honors. And at this time, he is very well funded from the NIH, from the VA and companies. And he probably doesn't even know that I signed checks from Gilead a number of years when I worked there, and we had a great collaboration on PAD 2 and PAD 4 in rheumatoid arthritis. Professor Robinson has about 240 publications altogether, many of them centered around rheumatoid arthritis, various aspects of the immunology of autoimmunity and RA, many of them around B cells, and in the last few years utilizing the new technologies of single cell RNA sequencing and mass cytometry and other more comprehensive ways of trying to elucidate what's going on in the joints of RA patients and OA patients as well. And so he is going to talk to us today about the origins of rheumatoid arthritis. So the microphone is yours, Bill.
Great, thank you so much. I appreciate the opportunity to virtually visit and to present at your Grand Rounds today. And I'm gonna talk about the origins and how people develop RA. I have one disclosure. And so I'll start by summarizing some background about autoantibodies in RA, and then describe to you our sequencing of the ACPA PlasmaBlast repertoire, and then single cell transcriptional analysis of ACPA and RFB cells. And I'll finish by telling you about some of our recent works identifying oral mucosal breaks as key drivers of RA development and flare. So in RA, the synovium becomes infiltrated with inflammatory cells. And, you know, we have come to appreciate that citrinated antigens are the targets in about 2/3 of RA patients, but people still don't know exactly which citrinated antigens are the key targets and the exact mechanisms by which they drive autoimmunity. Nor do we know how tolerance is initially broken against citrinated antigens and how these anti-citrinated protein antibody responses develop and evolve. And really that's been a center point of our research studies. Obviously, the two key prototypical antibodies in rheumatoid arthritis are rheumatoid factors, which target the Fc domain of IgG, as well as anti-citrulline protein antibodies that target these citrulline modifications on polypeptides. So when arginines get converted by the enzyme peptidyl arginine deaminase to citrulline, that's the target of these ACPA responses. This is a figure from a paper that we published almost 10 years ago now, showing that anti-citrulline responses first developed approximately 10 years before humans developed clinical RA. And so this is in collaboration with Mike Hollers and Kevin Dean at the University of Colorado. And this is from the US Department of Defense Serum Repository, where they collected blood every 2 years from individuals in the military. And then a bunch of people developed RA at this time point here, denoted by the red line. And this is thousands of days prior. So we're out now 10 years prior to the development of RA. These people are asymptomatic. And we profiled antibodies against these different citrulinated and also their native counterpart epitopes, all of which we had identified in established RA synovium by mass spec. And you can see people starting 10 years prior to the development of clinical RA started developing autoantibodies against these epitopes. And there's epitope spreading, and that crescendos together, comes together to ultimately give rise to RA. We also, my lab has also been very interested in the mechanisms by which citrination of specific epitopes on polypeptides results in RA. And we've obviously shown, based on the prior slide and as others have shown, that this creates neoantigens. But we also show that it turns the citrullinate protein into an autoadjuvant, that when you citrinate fibrinogen or histone or other molecules, it makes it bind TLR4 10-fold more potently and thus effectively dual stimulate. So we have this paradigm in which immune complexes with citrinated antigens dual stimulate macrophages by binding Fc receptor and TLR4, and that when you citrinate, for example, the fibrinogen, it binds the TLR4 10-fold more potently to more potently induce this dual stimulation and TNF production. We also show that citrination of, in this case, histone 2B in this ANR paper propagates netosis. And this is the classic model for RA development, where individuals at genetic risk, they have perhaps the shared epitope or a PTPN22 or other polymorphism, and they get exposed to certain environmental risk factors such as cigarette smoking. They get systemic autoimmunity, progresses to arthralgia, and then to unclassified arthritis, and finally to rheumatoid arthritis. And today I'm going to present you a story that ultimately leads us to the conclusion that oral mucosal breaks are driving this process along the way, from converting people at genetic risk into systemic autoimmunity and making them progress towards RA, and also promoting flare and perpetuation of established RA. And I'll provide you the data starting with initial sequencing and antibody characterization to defining the specific bacteria and the role of mucosal breaks in driving this process. So now I'm going to transition to the second part, where we're going to sequence anti-citrinate protein antibody PlasmaBlast repertoires and characterize the specificity and function of the encoded antibodies. So where do B cell responses arise? They occur because naive B cells and memory B cells are stimulated via the primary B cell response or a recall response to enter lymph nodes and germinal centers, where they undergo the process of affinity maturation, where they make daughter B cells that have been differentially mutated in their antibody binding domains, and they undergo niche competition for the B cells that make and encode antibodies of the highest affinity. And the B cells with the highest affinity antibodies receive growth factors and are stimulated to ultimately be released from the germinal centers, both memory B cells as well as plasmablast. And these plasmablasts, very importantly, circulate in the blood for several days and then ultimately deposit in tissues. But the point of this slide is that in our studies, we're drawing blood from human RA patients with active disease, and we're capturing the plasmablast. And thus the plasmablasts represent the ongoing immune response of the moment. And once an antigen trigger goes away, the plasmablast response goes away, for example, in vaccination. But in RA, these plasmablasts persist over time. And I'll show you evidence that the ACPA plasmablasts persist over a multi-year period of time from the same families, and that they're targeting these oral mucosal microbial bacteria. So this is just showing that if you look at disease activity in RA, this is just the standard stain for plasmablast, that there's an overall correlation between increased blood plasmablast and higher disease activity by the DAS score. Not perfect, but there's a general correlation. And so we've been isolating plasmablasts from RA and basically using initially our in-house sequencing methods and more recently 10x sequencing to sequence their antibody repertoires and their whole transcriptome, and then expressing their antibodies recombinantly to characterize their binding and functional properties. And when we do this, we also add in citrinated peptide multimers as baits. So we take these same antigens, many of them were on that antigen array that I showed you the profiling of the military subjects before. So these are all citrinated epitopes identified in immune complexes in human RA joints. We basically synthesize them as peptides and we multimerize them with fluorescinated avidin. And that lets us stain memory B cells or plasmablasts such that we can either stain them with fluorescent, or in the 10x mode, we use a avidin that has a 10x barcode, and thus we can sequence them directly without having to express the antibody to know their specificity. We already know because they bound our fluorescent or our 10x oligo bound multimer that they're ACPA B cells. So this is our workflow where we're taking these samples from RA, we're antigen labeling them with those citrinated peptide multimers that I just showed you. We're doing different sorting or 10x. And then we have one line of work where we perform paired heavy light chain sequencing, and we analyze their VDJ sequences and identify antibodies within clonal families and recombinantly express them and test them on arrays and determine their specificity and characterize their function. And then in a second line of workflow, we do single cell whole transcriptome analysis. And we look at the transcriptome of the B cells to understand the mechanisms by which ACPA and rheumatoid factor B cells have broken tolerance. So first, I'm going to present to you some of our work on sequencing specific antibodies within the B cell repertoire in RA and characterizing their specificity and function. And so just, when we sequence these antibody repertoires, I'm just going to show you, how we use clonal families to identify the B cells being generated from the germinal center response. So basically, what we sequence in the blood is these plasmablasts. And so they've emerged from their parent B cell, and these are the daughter B cells, the plasmablasts. And when we display them on a phylogenetic tree of the antibody repertoire, these green daughter plasmablasts from this original parent that have been activated are in this clonal family. And you can see the other clonal families of the blue B cells or the red B cells. And then these individual ones are called singletons. And so this kind of terminology, these clonal families, will come up repeatedly in my presentation, because that's how we identify the B cells that the germinal center has selected and for affinity maturation and is releasing into the blood, and that we're sequencing. So we're very focused on identifying clonal families of B cells based on their B cell repertoire to then express their antibodies and characterize their binding and their function. And in contrast to the old days, we now just choose a representative antibody and we just gene synthesize the heavy and light chains, and then we just transiently transfect it, and a month later we have a recombinant antibody that we can characterize in the lab. So with Mike Hollers, we sequenced his preclinical RA patients. So these are patients with who do not have RA, they're asymptomatic, but they have a first-degree relative with RA. And these individuals with a first-degree relative are asymptomatic and they're CCP positive. And we sequence their repertoires. And what we find is that they have all these different clones that bound to our citrinated peptide multimers. All the colored clones bound our citrinated peptide multimers. So these are ACPA B cells. And further, we have multiple clonal families of ACPA B cells in which there's both IgA and IgG members. So this is a clonal family that bound to our citrinated peptide multimers. The red are the IgGs, the teal are the IgAs, and you can see multiple different families, all with interspersed IgG and IgA members. Further, we show that there's a skewing towards IgA plasmablast in these preclinical patients. So these are patients who are asymptomatic, they're CCP positive, but they have a first-degree relative with RA. So they have like an 8% risk in their life of developing RA. And you can see they have a skewing or increased level of IgA plasmablast as compared to healthy or early RA. And further, when we determine clonal lineages of B cells based on their B cell receptor, we see them persist over time. So these lineages of IgA in light blue and IgG in purple, and these are different clinic visits on the order of 3 months apart over kind of a year time frame. These plasmablast clonal families, including both IgA and IgG, are persisting over time in these patients. These are the ACPA plasmablasts. And further, we see the same result in multiple other patients, that they exhibit plasmablast, ACPA plasmablast, where the lineage persists over time. Normally, if an antigen stimulus goes away, like in the flu vaccine or in a staph infection, the plasmablast will come up during the acute response to the vaccine or the infection and then dissipate on the order of 5 to 10 days following resolution of the antigen from that vaccine or infection. But in the case of RA, we're seeing the persistence of those plasmablasts over a year-long time frame, which suggests persistence of antigens that's stimulating them. This is just showing that we see trafficking of these ACPA plasmablasts from the blood to the synovium. And further, and interestingly, when we look at the heavy chain of these plasmablasts, this is the number of mutations from germline. So how much affinity maturation an individual plasmablast has had. And this is the median levels. You can see higher levels in the blood as compared to the synovium for heavy chain, meaning the ACPA plasmablasts are more mutated in the periphery than they are in the synovium. And likewise for the light chain, they're more mutated in the periphery or the blood as compared to the synovium. But when they traffic to the synovium, the plasmablast expands. So this is the size of the clonal family, and it's basically smaller in the periphery and expands when it goes into the synovium. And so we interpret these data to suggest a periphery-driven question mucosal because of this IgA co-clustering and IgA predominance response. It's just showing that when you look at the number of mutations, this is in the heavy chain of these ACPA plasmablasts, or actually ACPA plasmablasts on the left, non-ACPA plasmablasts on the right, and the light chain, the same thing, that the ones that are reactive with the CCP in the CCP assay are more heavily mutated than their non-citrulline reactive plasmablast counterparts, suggesting that this affinity maturation is giving rise to their anti-citrulline reactivity. This is just showing an example of a clonal family. This is a clonal family of ACPA plasmablasts. This is their heavy chain. This is their light chains. And this is their amino acid sequence, and the CDRs are boxed. And basically what all the color changes are amino acid substitutions. And this level of affinity maturation in these heavy and light chains from these clonal family plasmablasts, this is a massive amount of affinity maturation. Normally, even in antigen stimulated like flu vaccine, it's an order of magnitude less affinity maturation than this. This is like HIV-like in the degree of affinity maturation. It's estimated that a B cell accumulates approximately 1 nucleotide mutation per trip through the germinal center. And to get amino acid substitution, you have to have more than 1 nucleotide, because not all single nucleotide alterations result in an amino acid chain. So basically, these plasmablasts in this clonal family have gone through the germinal center over and over and over again to achieve this degree of affinity maturation. So we next, this is just showing that this observation, the prior slide was this clonal family here, and clonal family 2 and 3 exhibit a similar degree of massive somatic hypermutation. And so we express the antibodies indicated recombinantly to characterize their binding characteristics. And we show that from this clonal family 1, the one that I just showed you the sequences for in the last 2 slides, that the different members that are divergently mutated have different binding reactivities for these citrulinated epitopes. For example, this antibody 60 from the clonal family, they all came from the same original parent B cell, bind citrulinated vimentin, but to a much lesser degree, citrulinated fibrinogen. Whereas this antibody 78 that's differentially mutated, but within the same clonal family, binds citrulinated fibrinogen and less citrulinated vimentin. So this basically shows, and we see similar observations with other clonal families. So this basically demonstrates that this extensive affinity maturation is resulting in epitope spreading, the divergent reactivity of different B cells against citrulinated antigens. This is just tracking how the B cells got there. So this is the predicted germline. These are the B cells we sequenced in the blood. And you can see the numbers of nucleotide or amino acid substitutions they had to, actually these are nucleotide changes that they got to get from the germline to what we sequenced in the blood. Literally, 100, tens to 100 plus mutations. And this just shows similar observations for other clonal families, indicating this massive amount of affinity maturation that can only occur through multiple trips through the germinal center, suggesting a persistent mucosal drive. And I'm going to tell you later on that we think this is an oral persistent mucosal drive due to mucosal breaks. This is just showing that when we take our recombinant antibodies, that they bind to citrulinated but not native human antigens, and that it's associated with mutation level. So this is the mutation level here, and it's the more heavily mutated, the monoclonals that bind the human citrulinated antigens, whereas the lesser mutated RA patient plasmablast monoclonals do not bind the citrulinated targets. Okay, just one slide on function. Just we can take our monoclonals and we can form immune complexes with them. This is showing, this with citrullinate H2B immune complexes. It's triggering neutrophil netosis based on cytox release. So these antibodies can be functional in terms of eliciting a response. So just to summarize this part, we believe it implies this is our first evidence for perhaps a mucosal initiation and drive of these ACPA responses in RA. We saw these IgA plasmablasts that encode ACPA. They class switch to IgG. And in established RA, we see not only in preclinical, but also in established RA, we see persistent IgA plasmablast over a kind of a 1+ year timeframe. We see increased somatic hypermutation in the periphery as compared to the synovium, suggesting a peripheral drive. But we see clonal expansion where the cells traffic to the synovium. We show that these recombinant ACPA can dual stimulate macrophage TNF production, can induce netosis. I didn't show you this. This is in our published data. Can induce netosis. And we need to obviously further define their roles. So I'm next going to just show you a few slides on some of our single cell transcriptomics that show different mechanisms of tolerance breakdown in ACPA versus rheumatoid factor B cells. So this is that second line where we're doing single cell transcriptomics in ACPA and rheumatoid factor B cells. And we basically sorted and used 10x oligos on antigens to label and sequence RF B cells, ACPA B cells, and basically tetramer negative B cells. And we sequenced naive, memory, and plasmablast based on our transcriptional classifiers. And then we informatically compare them. And this is just showing that in ACPA and RF B cells have distinct transcriptional programs at the single cell level. ACPA B cells have very prominent germinal center response and B cell activation response, suggesting that they're kind of actively going through the germinal center, which is consistent with those lineages being persistent in blood over a kind of many months to a year plus time frame that I showed you a couple slides ago. In contrast, rheumatoid factor B cells have a proliferation, activation, and an innate immune response signature that I'll show you a little bit more in the coming slides. So just ACPA plasmablasts, germinal center response. And we also see this evidence of breaking tolerance in the primary response for ACPA B cells. And this just, ACPA B cells exhibit this primary antibody response signature. So in addition to this, in addition to the ACPA plasmablasts that we're seeing, we also see B cells that bind to our citrinated multimers. So they're ACPA B cells, but they classify as naive B cells. And thus we think that these are ACPA primary response B cells. And I'll tell you a little bit more about these in a minute. So these are these ACPA primary response B cells, and basically they classify as naive B cells based on their gene expression profile. And they have increased JAK3 and then a bunch of class switching factors, MSH5 and XOC4, suggesting that they're preparing to go into the germinal center. They also have decreased inhibitory receptor signaling. And I would note that these naive classifying activated ACPA B cells were also seen by Dana Orange in her New England Journal of Medicine paper as preceding actually the prime cells in her fingerstick RNA-seq studies, in which she studied, which she shows the B cell response and then prime cells and then RA flare. And her B cell response was actually these naive activated B cells. And we believe these are the same as our ACPA naive B cells. And so we believe this suggests that tolerance could be broken in the ACPA B cell primary response. And so thus we see dysregulated ACPA B cell activation at 2 levels. Obviously there's this huge ACPA plasmablast trafficking through the germinal center, but we also see this ACPA B cell primary response that's activated and prominent in established RA, which was also seen pre-flare in the Orange et al. New England Journal of Medicine paper. So we're very interested in this activated primary response ACPA B cells. Rheumatoid factor B cells, in contrast, exhibited a very prominent innate immune response signature characterized by MyD88 signal, interferon gamma signaling. And so in contrast to the ACPA B cells, the rheumatoid factor memory B cells have this very prominent innate immune response that are likely being activated by immune complexes. So just to summarize, these ACPA and rheumatoid factor B cells, ACPA B cells, there's a very prominent plasmablast signature, highly mutated with tens to 100 plus mutations, with a very prominent germinal center signature. In addition, there's a second ACPA B cell primary response with these kind of class switching genes and activation genes, but yet they haven't affinity matured yet. And then the rheumatoid factor B cells have this innate immune response signature. And this is just summarizing those two together. There's this ACPA B cell primary response, the ACPA plasmablast response that have heavily affinity matured, and then these rheumatoid factor B cells that are likely internalizing citrinated antigens and promoting B cell help to the ACPA B cells. So I want to kind of spend the last 10 minutes or 15 minutes talking about mucosal breaks and their potential role in providing the basis for everything that I just presented, like being the antigen stimulus for all of this. And this was one of our first observations. We started, we basically took all our monoclonal antibodies from RA PlasmaBlast, and we also took RA plasma and synovial fluid IgG. And we show that they bound to citrinated bacteria as well as to human citrinated antigens. So here's that same panel of RA monoclonal antibodies. So these are monoclonal antibodies from RA blood plasmablasts, activated B cells. And we're putting them on our antigen arrays, in which we put on our antigen arrays either human antigens, citrinated at the top across these rows, native, or gingival bacterial antigens, citrinated bacteria, native bacteria. These are the bacteria listed over here, citrinated native, or periodontitis bacteria, citrinated up here, native at the bottom, or gut bacteria, citrinated at the top, native at the bottom. And we're looking at binding. And you can see basically our heavily mutated RA blood plasmablast encoded antibodies bind to human antigens, citrinated, but not the native. And they bind to both gingival and periodontal and to gut bacteria, all when they're citrinated, but not in their uncitrinated forms. In contrast, the less mutated antibodies didn't bind to any of these targets. Here we're showing, sorry, my slides are jumping. Here we're showing, so we're extending this slide is of our monoclonal antibodies from individual plasmablast in the blood. Now we're using sera, or at the bottom, synovial fluid from RA patients. And we're using the same bacteria, citrinated, native, citrinated, native. These are just different gingival bacteria, Strep parasanguinus, Strep borealis. You can see increased binding of RA blood antibodies to the citrinated Strep parasanguinus compared to, just deconvoluting. Healthy control. So the gray is the healthy control, the blue is the RA. And the native bacteria, they're similar. And here's another oral bacteria, citrinated, when we in vitro citrinated, Strep borealis, higher with the RA compared to the healthy control. Native doesn't make a difference. And then periodontal bacteria, this particular one did not exhibit a difference. Gut, also when you citrinated, it's higher down, GU, and we're here gingival. Now we're looking at synovial fluid, and it's the same trend. So basically, multiple oral bacterial, if we citrinate them, they exhibit higher reactivity to RA serum. Certain other bacteria from other sites, like this is an example in the gut, also exhibit higher reactivity when you citrinate it. But there's also certain bacteria that don't. So we needed to kind of further deconvolute this whole story, and we show we're going to hone in ultimately on the oral bacteria as we think as key drivers in a subset of RA. This is just showing bacteria. When we isolate bacteria out of the mouths of humans with periodontitis, so that's deep-seated infections in the gum, that many of their bacteria are citrinated. And this is just looking at the percent citrination in live bacteria when we take bacteria from the mouth, from the gut, or from the vagina. And there's the highest degree of citrination in the mouths of, this is patients with periodontitis. We perform mass spec analysis of the oral swabs from people with periodontitis, and we identify a bunch of citrinated targets. We can put those on antigen arrays, and we show now our RA monoclonals are in this vertical as opposed to the across the bottom, but RA monoclonals here. And you can see they bind to several of the different citrinated oral bacterial epitopes, but generally not to the native versions of them. Sorry, I think something's on my keyboard that keeps flipping it. Whereas a bunch of antibodies didn't bind to either. So it's a specific set of RA PlasmaBlast antibodies bind the citrinated oral bacterial epitopes that we identified by mass spec in brushings of oral bacteria. Okay, we collaborated with Dana Orange and looked at her fingerstick RNA-seq, and we basically extracted non-mammalian reads, and we found a bunch of bacterial reads. And this is just looking at abundance of bacteria on the oral. You could see there's an uptick here in the skin, looking at different classes. And this is looking in the blood fingerstick samples for evidence of bacteria and showing that we see significant elevations in RA patients of bacteria from the mouth. So from the tongue dorsum, classic kind of bacteria that reside there, supragingival plaque, buccal mucosal, but not from the interior nares, not from the behind the ear or the gut or the genital tract. We also show, working with Dana Orange, that there's repeated episodes of oral bacteremia. So we're looking at different oral bacteria, and we're seeing oral bacteria spike in the blood of RA repeatedly. And this is just one patient's tracing. So this is on their weekly fingerstick RNA-seqs. And this is a second patient's tracing. And there were several patients with dental work where it dramatically exacerbated this, but they were also exhibiting repeated oral bacteremias that were asymptomatic, completely independent of any dental work. We show that in the human RA synovial gene expression data sets, that there's an oral defense and innate immune response pathway. So basically, in the high inflammatory RA, they exhibited a oral defense and innate immune response pathway, suggesting that some of these cells may be trafficking to the synovium. So I just have a couple summary slides, and I'll finish up in just in the next 3 minutes or so. So just to summarize the last sections, we believe our data suggests that in patients with periodontitis, they have these mucosal breaks that release oral bacteria into into the tissue, and they become citrinated by host neutrophils, and that these then elicit a PlasmaBlast response and ACPA that then promote joint tissue destruction in RA. And that in multiple RA patients that we studied, they're having repeated asymptomatic transient oral bacteremias that we believe are stimulating repeated rounds of ACPA PlasmaBlast activation and repeated rounds of cycling through the germinal center and affinity maturation, resulting in these ACPA antibodies that bind to human citrinated antigens. So taking that origin of RA slide that I presented at the start, this is how we would use our data to change that that slide. We believe that mucosal breaks, particularly oral ones, are driving both the initiation of autoimmunity in genetically susceptible individuals, the progression of autoimmunity and the development of RA, and the persistence and progression of RA. We believe that other mucosal breaks, such as in the lung or in the gut or in the genital tract, might also contribute, but our data is specifically showing a key role for oral breaks in at least a subset of ACPA positive RA patients. Just recognize the people in my lab: Camille Brewer, Nitya Ramadas, Sarah Kompethith, Alejandro Gomez really drove this work. And we work in close collaboration with Mike Hollers and Kevin Dean in Colorado, as well as with Dana Orange at the Rockefeller. And we collaborate also quite extensively with Jane Buckner and Eddie James at the BRI. So with that, I'll finish, and the floor's open for questions.
Thanks very much, Bill. That was an incredibly interesting presentation you gave, and there are some questions already, and I have a ton of them myself. So I'm going to start with a question from Keith Elcon, who asks, are the properties, oops, I lost it here. I lost the chat, so maybe Keith, if you...
Here I see his question. Are the properties of IgG rheumatoid factor more similar to IgG ACPA?
There we go.
Thanks for that question, Keith. We find that they're actually completely different transcriptionally. So when we look at the memory, when we look at memory B cells that encode rheumatoid factor, they're very innate immune response signature, and they have very low mutation levels in their rheumatoid factor antibodies. Like they have like less than 20 nucleotide mutations. Whereas when we look at the same corresponding IgG ACPA memory B cells, they have that 100 nucleotide mutations, and they have that germinal center transcriptional response. So we actually find that they're quite different in their profiles. But obviously, it'll be interesting to see, we think, my lab thinks rheumatoid factor is a very important contributor to RA pathogenesis and a very important potentiator of ACPA responses, because we think it conglomerates ACPA to potentiate macrophage activation and immune complex mediated destruction. We have an A&R paper from like 5 years ago suggesting that, or providing data to support that role for rheumatoid factor.
Can you, Bill, see the Q&A box?
Yeah, I'm looking at it right now. Yeah, I mean, I think, we've long known that periodontitis has been a risk factor for RA, or it's been hypothesized to be a risk factor for RA. And there's some epidemiologic evidence, meaning if you test all people with periodontitis, which means deep-seated pockets and or infections in their gingiva, that 4% of those people are anti-citrulline protein antibody or ACPA positive. And so, our studies are providing a mechanistic kind of basis for how periodontitis could be associated with the development and the progression of RA through these mucosal breaks of bacteria that, and importantly, our data show that it's not endogenous bacterial pads, but it's likely the host neutrophil pads that are citrulinated the bacteria. So this concept that you need to have an endogenous pad, we don't think is, that may be the case for certain bacteria, but we think these bacteria are getting citrulinated by the neutrophil netosis in the host pads.
So I have a question, Bill. Given that you're proposing a role for bacteria. Wouldn't you think that an anti-TNF treatment would be detrimental for defense against such bacteria, and that you might have an exacerbation of the periodontitis and worsening of things?
Yeah, no, in general agree with what you say. And we wrote a editorial with Mark Genovese and Larry Moreland suggesting that the reason why anti-TNF is associated with MS is because it aggravates autoimmune responses, but because anti-TNF can't get into the CNS, it can't block the downstream mediator. But in the case of RA, even though it may aggravate the autoimmune response, anti-TNF, we think aggravates the autoimmune response, it blocks the downstream mediator. So ultimately all is good because you block the tissue destructive cytokine, right? But I think that's why when you take your patient off anti-TNF, the RA comes back raging. Like it's, when my patients come off anti-TNF, the RA comes back like gangbusters.
So would you also propose then that the T-cell acting therapies like abatacept work mostly in the germinal centers by reducing B-cell somatic hypermutation and so forth?
Yeah, I think it could very much, impact those. We've been working with BMS trying to show that, and it hasn't been as easy to show as one might think. In converse in cancer, we show that ipilimumab, which is the converse of abatacept, because it's potentiating signaling through CTLA4 or CD28 to treat cancer. We show that increases plasmablast, but we've had, anything that really reduces the activity of RA reduces the plasmablast response. If you really effectively block RA through, abatacept, TNF, JAK inhibition, Ritux, the plasmablasts go down in all those situations. I guess in Ritux, you're depleting the precursor, so maybe that's not a fair comparison, but.
Yeah. So here's an interesting question from Mark Wenner. The gender difference in RA, might it be because of vaginal mucosa participating in pathogenesis?
Yeah, I mean, Kristen Demerel at Colorado, working in Mike Holler's greater group, she's busy looking into those things. And she's also sending my lab B cells from breast milk, the sequence their repertoires from RA patients. So they're busy running that to ground. I mean, I don't think our data, just to be clear, our data clearly implicates the mouth in a subset of RA, but we don't exclude, like you could see, we citrinate the gut antigens and they react too. So gut mucosal breaks or other mucosal breaks could also play a role. It's just we didn't we were focused on the mouth. And that's what our sequencing data showed. Like we looked hard for gut and genital and other breaks in our sequencing data, and we didn't see them. We just saw mouth ones,
So there's one more question from Bobby, and I had the same question myself. Why the joint? Why do all these events lead to inflammation in the joint, of all places?
I mean, that's the age-old question, but it's the same thing in lupus. Like, why the kidney or why the, and you could come up with explanations. I mean, the classic explanations for why the joint, it depends who you ask, right? Some people will say it's because the joint has acellular surfaces, the cartilage. And therefore, if you get deposition of immune complex, you don't have membranes and thus the normal complement inhibitors. If you ask Christophe Benoit and Diane Mathesis, they would say, and they published in Nature Immunology, that there's different permeability of the vasculature of the joint compared to other tissues and organs. And that's why their KRN immune complexes go to the joint and cause joint inflammation. I think there's just unique properties about the cellular and structural features of the joint, both in terms of having acellular surfaces where collect immune complexes without the natural complement inhibitors perhaps vascular differences that better allow their penetration as well as immune cell penetration. And perhaps Dana and I actually think that we're very interested in like micro trauma, right? You know, like you cruise around and you bump your joint, and like maybe you get a micro bleed or some micro trauma. Maybe that causes some citrination there, and that kind of is the spark that ultimately lights the fire. And there's also OA in the joint. RA is, as most people know, it's bimodal in incidence. There's one peak at age 30 to 50, and then a second peak rises until age 80. So maybe OA is also a joint injury that, you know, has a very low risk, but a slight one by causing damage that maybe citrinates things and causes injury that ultimately, so the question is like, why do all these people have ACPA, but they don't have RA, and then they convert to RA, right? And so, you know, it's probably some sort of features of the joint that all of a sudden make that ACPA promote inflammation and then recruit this adaptive autoimmune response.
So I still have one more question about the citrination itself. There, at least in the early literature, there was much to do about the increased citrination, where by histology and other methods, folks could document an enormously increased citrination of lots of proteins. How does that factor into your thinking about the early initiation of RA?
I think it's tricky because there's a ton, you have citrinated proteins in every cell in your body. In the nucleus of every cell in your body, there's like citrination of histone that winds your chromatin. In your skin, like there's citrination. In your myelin, there's citrination. That's doing normal physiologic good things. Yeah, I'm not sure. It's a, you know, there's obviously this component of a HLA and other genetically predisposed host in the context of this citrination. So I think that's, you know, like, why do we all have citrination in every cell in our body, yet only 2/3 of our RA patients are truly autoimmune against it, right? I think that's a great question.
Yeah. We'll discuss that more when we meet later on. Yeah. So are there any questions out there that we haven't asked yet? I'll give people 30 seconds to type something. It looks like Mark Wenner has one more question here. So the serial visit data figures you showed looked like clonal families present at visit 1 and 2 were gone by visit 3 in healthy at-risk relatives. Would you comment on that?
Yeah, I think, you know, we clearly see persistence of sets of IgG/IgA mixed clones over like a prolonged period. And they're ACPA, and they bind these citrinated bacteria and human antigens. So, and then separately in our sequencing study, we see these like recurrent episodes of oral bacterial DNA in the blood. And when we do the brushings, they're citrinated, and they're the same ones these ACPA target. So, you know, I don't think it's watertight that at every moment the RA patient necessarily has that exact same response, but I think they're getting the same stimulus over and over and over and over again. And that's what we believe is resulting in these massively mutated ACPA that cross-bind both the oral bacteria and the human target. I mean, yeah.
So maybe a final question. Do you think there's a place for antibiotics or, you know, a serious discussion with the dental community about all of this?
I think it is. And, you know, there's a bunch of trials ongoing, particularly in Europe, trying to do different things in the mouth. But yeah, I think, and if that's really the case, that would be a fundamental approach. I don't necessarily think anybody who thinks all of RA is one disease or one trigger is definitely off base. So, I mean, I think this is like 25% of RA patients, I think, could have a real oral trigger. So, but yeah, it would change. Like if we're right, then it could really change how you do things. So, yeah. Like in the way you would fundamentally treat them. It's not about giving them a Batacept or TNF or whatever. It's got to help, you know. I always tell my patients, make sure they're brushing their teeth and going to the dentist, like, you know, rigorously.
Maybe it's too late.
Right. Well, and that's the, so if you're a conspiracy theory person, which I'm generally not, but the conspiracy theory for smoking is all smoking is, everybody thinks smoking causes RA, but maybe smoking, all it's doing is causing you a periodontitis, because they're inseparable. If you smoke meaningfully, you have periodontitis, right?
But not all smokers get RA in the end.
No, for sure. But like, you know, just like, yeah, I mean, that's the whole conundrum, right? So,
yeah. All right. Well, thank you very much for a wonderful seminar and a great Q&A in the end. And I'm looking forward to discussing with you a little bit later today.
Yeah, it's fantastic. Thank you so much. Appreciate the
invitation.
invitation.
Thanks.
Okay.
Bye-bye.